Hi students, in this class we have to talk about the regulation of gene expression. So far we have studied how far the DNA becomes, that is what we have the gene. Then how the DNA stores the genetic information, how the genetic information is transferred to or expressed. But in this class we have to talk about the regulation of gene expression. One of the important aspects, how far the gene expression is regulated. Either it is by some other gene or any other products of the body. Now, the regulation of gene expression has been studied extensively in the case of prokaryotes, particularly in the case of E. coli. Now, normally, the gene expression occurs at different levels during the process of what we call transcription or translation, or we have different stories. Now, what are the stages at which the gene regulation occurs? The gene regulation occurs either at the transcriptional level, or we have that is at the processing level, This is otherwise called post-transcriptional level. Or during the transport of mRNA. From nucleus to cytoplasm. The transport of mRNA after the formation, they are transported from nucleus to cytoplasm, or at the translational level. So the expression of the gene is regulated at these four different levels, either at the transcription level or at the processing level. There is nothing but what we have the splicing process, or we have the transport of mRNA from the nucleus to the cytoplasm, or at the translational level. These are the four levels of gene expression regulation. That is in the case of eukaryotes. There are four regions or four just actually level. So in the case of eukaryotes. So the expression of gene regulation occurs at four levels. But if you are taking prokaryotes, the regulation of gene expression, the expression and control, that is normally regulated, or the gene expression is regulated and controlled only at the transcriptional level. This is in the case of prokaryotes. And there is no processing, as the genetic components are present in the cytoplasm without any nuclear membrane. Now the regulation of gene expression occurs only at the transcription level in prokaryotes, but in the case of eukaryotes, in all these four levels, any one of these four levels, we have the regulation of gene expression occurs. Now here in this chapter, we are going to talk about the gene regulation or regulation of gene expression only at the transcription level. That means we are talking about only the prokaryotes, not in the case of eukaryotes. If you study this one, that is in the case of prokaryotes, you can understand how far the gene expression is regulated in the case of eukaryotes. Now, we have normally some metabolites are formed, which are smaller in size. Some metabolites, which are normally smaller in size, produce either extracellularly or intracellularly. They trigger what we call the initiation process or inhibition process of what is called the gene expression. Smaller molecules produced extracellularly or intracellularly can initiate or inhibit the gene expression. This is one we can say any metabolites. Normally we can say. Now what do you mean by actually Operon? So here we have to talk about Operon. What is Operon? Now the gene expression is regulated by one unit, what is called operon. Now what is an operon? A cluster of genes that normally with related function is called as an operon. What is an operon? It is nothing but a cluster of genes with a related function, a short accurate description. So while it is being transcribed, it transcribes only one mRNA molecule. We'll discuss later. If you're taking E. coli, Escherichia coli bacteria, there are nearly about approximately 260 genes. These 260 genes are grouped into nearly 75 different operons. This is one what is called the statistical data. Now let's take the structure of operon. So it is nothing but a unit, what is called actually responsible for transferring the genetic information. Genetic unit consists of one or more structural genes. Number two, adjacent to the structural genes, we have a site, a promoter site. That is the one responsible for the initiation of transcription. And also an on-off switch, that is what is called operator site. When the operator site is on, now the process of transcription occurs. When the operator site is off, that is the transcription is normally stopped. So it is acting as on-off switch. So I represent in a simple diagram. Let us take an operon, simply any operon. I am taking only an operon. So an operon consists of, we have structural genes, one or more structural genes. So in the case of prokaryotes, we are talking about the prokaryotes, the regulation of gene expression at what is called the transcription level. The gene is polycistronic. Let us take three genes, the structural genes, A, B and C. They represent the structural genes. They code for either protein or RNA, tRNA. Now adjacent to this structural protein, we have a region. This is the region. So this is what's called the promoter site. Promoter site. And this is acting as on-off switch. When it is on, the transcription occurs, leading to the formation of mRNA. So this is mRNA, which is being transcribed by the structural genes. So initiation. Okay. So when it is on, then what will happen? The three genes are transcribed to get what's called a single mRNA. Now nearer to the promoter region, we have another region, what is called, So this is what we have, just I want to take another one. This is the operator gene, not the promoter gene. I mentioned about this operator gene, just O. The operator gene, which is acting as on-off switch, that normally responsible for the transcription of structural genes. If it is on, we have transcription occurs leading to the formation of mRNA. Now next to the operator region, we have what is called a promoter. That's the main region, because the RNA polymerase is found attached to only this site, what is called as a P site. What is called as a P site? The promoter site. The RNA polymerase responsible for the transcription of these genes. For example, the polycistronic genes. We have three cistrons, hence called polycistronic genes, as in the case of prokaryotes. So all the genes are being transcribed only when the RNA polymerase binds to the promoter site and moves along the O, that is the operator site, and reaching the initiation process site, or we have the transcription. So we have an operon consists of one or more structural genes, and adjacent to it we have the promoter region, and in between the promoter and structural, that is the genes, we have an operator. It is on-off switch. The RNA polymerase binds only to the promoter region and moves along, that is through, just we have the operator region. If it is blocked by somebody, there is no transfer of RNA polymerase to the site of transcription, to the initiation site. So if it is open, just like a railway gate, if it is open, then there is a passage. it is blocked, there is no passage of RNA polymerase directed by the promoter region to reach the initiation site, so there is no transcription. So that there is no formation of a specific protein. So this is what we a simple one, I am saying that one. Now what are the components once again? So simply a definition is given in the book. It consists of one or more structural genes, an adjacent promoter site, and also we have the operator site. Now, what is the role of the structural genes? The structural genes code for either tRNA or mRNA or proteins. Now, what do you mean by that is promoter region? It is a signal regions having signal sequences of DNA. Signal sequences. This promoter region consists of signal sequences that initiate the process of transcription. The signal is coming only from the promoter region, then we have the switch is on or off, and that is the signal sequences of DNA, which is called as a promoter region, and that initiate the process of transcription. Now the RNA polymerase enzyme responsible for the synthesis of mRNA binds to, that is what is called the promoter region prior to the transcription. Now we have the operator. The operator is present between what is the structural genes and the promoter region. It is on-off switch. It may be blocked or may not be blocked. If it is blocked, there is no transcription process. If it is not blocked, the process of transcription occurs. Okay? Sorry. We have another protein, what is called repressor protein. Repressor protein. Now this repressor protein binds to what is called the operator gene, blocking the passage of RNA polymerase to reach what is called the initiation site of transcription. That's why I say it is on-off switch. Now this operator gene O is normally just actually blocked by binding of repressor protein to it. It will block the passage of RNA polymerase to reach. If the repressor protein is not actually binding with what we call the operator gene, the process of transcription occurs. So the repressor protein always binds with the operator gene. The RNA polymerase binds with what we have the promoter region. So I am taking example of what is called the Lac Operon in E. coli. The Lac word refers to the lactose metabolism. So Lac refers to what is called lactose. The operon that is concerned with the lactose metabolism is called Lac Operon. Now for lactose metabolism we need 3 enzymes. What are the 3 enzymes? Number 1, beta galactosidase. Beta galactosidase. Number 2, permease. Number 3, transacetylase. Transacetylase. So these are the 3 enzymes responsible for the metabolism of lactose. Now the beta galactosidase is responsible for the hydrolysis of lactose sugar into, so lactose into glucose and galactose. There is a role of beta galactosidase. The another name for lactose is beta galactoside. That's why the name is given beta galactosidase. Another name for lactose is beta galactoside. That's why the name beta galactosidase is given for the breaking of lactose into glucose and galactose. The permease. This is an enzyme responsible for the entry of lactose into the cell from outside. Permeability. Increases the permeability for lactose sugar. Allows the entry of lactose into the cell. Transacetylase. It is responsible for the transfer of acetyl group from coenzyme A to beta galactoside, that is a lactose. In the book it is given beta galactosidase, it is not. Now it is responsible for the transfer of what is called acetyl group. That is acetyl group from the coenzyme A. That's why it's called acetylation process. As it transfer the group, acetyl group from coenzyme A, this is called acetylase. So trans-acetylase. These are all the three enzymes responsible totally for the lactose metabolism. Now, in the case of Lac Operon, these three enzymes are produced by three different genes. You know that one, in the case of prokaryote, the gene is polycistronic. That is, one promoter directs the transcription of just one or more genes, which results in the formation of one mRNA, which is translated into one or more polypeptides. That is the nature of polycistronic. So, one promoter, many cistrons or structural genes, one mRNA, resulting in the formation of what we call many polypeptides. That's the nature of polycistronic gene. Now, the prokaryotic gene is polycistronic. Now, a molecular model of this Lac Operon in E. coli was given by two scientists, one Jacob, another one Monod. In 1961, two people, 1961, Francois Jacob and Jacob Monod. So the Lac Operon model was proposed by Francois Jacob and Jacob Monod in 1961. I mentioned already here in the case of prokaryotes, the gene is polycistronic. Now there are three structural genes. These three are the structural genes. Three cistrons. Now these three actually genes are responsible for the transcription of messenger RNA, which translated from three different proteins, namely the enzymes. Now we have the genes called by name LacZ, Lac and Lac A. These three together form the structural genes, polycistronic gene. The gene Z is responsible for, that is having the code, or it encodes for the enzyme beta-galactosidase. Now the Lac Y gene encodes for the enzyme permease, and the Lac A encodes for, that's coding for, that is the enzyme trans-acetylase. So three different actually polypeptides are produced, the enzymes are produced from what is called the apron. That's what we call the polycistronic gene. But the mRNA formed is only one, that is called polycistronic mRNA. So these genes undergo transcription to form a polycistronic mRNA, which is then translated to form the three different enzymes what I mentioned. Beta-galactosidase, the permease, and also what we have, that is trans-acetylase. But under normal conditions, when there is an availability of glucose sugar, what will happen? There is a gene, what is called I gene. This gene is called what is known as the regulator gene. This is the regulator gene. Now, why is it given what is called I? I refers to inhibitor gene. It inhibits a process, what is called the operon transcription. That's why it's called inhibitor gene. Under normal conditions, when the source of energy is glucose, what will happen? The I gene undergoes transcription process to form a repressor mRNA. This is repressor mRNA. This is the transcription process resulting from the I gene inhibitor gene, leading to the formation of what is called repressor protein after translation under normal conditions when glucose is the source of energy. Now this repressor protein, why is it called repressor? It prevents or inhibits the activity, hence called repressor. Okay, inducer induces, repressor suppresses. Now this repressor protein binds with the operator site and thus actually blocking the RNA polymerase to move through it. So it is mentioned already on-off switch. While the repressor protein binds with the operator, what will happen? Now the switch is off. It doesn't allow the RNA polymerase bind to the promoter. Only when the RNA polymerase binds to the promoter region, it passes through the operator, then only the transcription occurs. But here, because of the block, the repressor protein binds with the operator, there is no movement of RNA polymerase along the operon. So here repressor binds to operator region and prevents RNA polymerase from transcribing structural genes. No transcription. So that's why I mentioned here, you see that one, there is no structural genes. So this is what is happening when there is actually glucose sources available. Suppose if the carbon source, glucose is not available. If lactose is available, then what will happen? The process of transcription happens. In what way? So we have to go for the next part of this one with another diagram. So here, formation of repressor protein binds with the operator gene. So there is no binding of RNA polymerase with the promoter. No transcription occurs. This is the reason why, what I given here. So that's why I mentioned here, in the absence of inducer. Who is the inducer? Somebody is needed to induce a process of transcription in the Lac Operon. So in the absence of inducer, it is happening. Now what will happen in the presence of inducer? Okay, now let's see how far the three enzymes are produced. I mentioned already, in the absence of lactose, when glucose is available as an energy source, the I gene produces what is called a repressor mRNA by transcription, which is translated to form a repressor protein which binds to the operator site and preventing the binding of RNA polymerase to the promoter, so preventing the transcription process. Now in the absence of the carbon source, what we have the energy source as glucose. Now if lactose is available, the lactose now binds with what we have the repressor protein produce. Binds with the repressor protein and causes a change in configuration of the protein, so that this repressor protein is no more able to bind with what we have the operator site. Now the switch is on. Now the RNA polymerase binds to the promoter site. It is moving along, passes through the operator site. Now the switch is on and initiating the process of transcription. So the transcription is started. We have the formation of what is called messenger RNA. This is called Lac messenger RNA, as it is normally carrying a message for the metabolism of lactose. This is called Lac mRNA, which undergoes the translation process. We have received the three different enzymes by Z beta galactosidase, by Y permease, by A transacetylase. All these three enzymes are taking part in the process of metabolism. Okay, this is what is happening when the lactose is present. Now here, the lactose is acting as an inducer, which stimulates the process of transcription. That is why the Lac Operon is called inducible operon. Inducible operon. Because the operon is induced by lactose, the inducer. When lactose is present, then we have the process of transcription. That's why the lactose is called inducer, and that is called inducible operon. Now this type of regulation of operon by a repressor, see the repressor is the one which regulates. This type of what is called the regulation of operon by what is called repressor is an example for negative control of transcription initiation. A negative control of transcription initiation. So this is an example for negative control, because the repressor prevents the transcription process. When inducer is present, what we have the process occurs. So this is called negative control of transcription initiation, as it starts transcription. So this is about. We also have, in the case of Lac Operon, positive control is also seen. Now if we are taking another operon, what is called tryptophan operon. Another operon, what is called tryptophan operon. I'll tell you only, I am not going to explain with the help of diagram, simply another example. tryptophan, another amino acid. Tryptophan operon. We have actually in the case of tryptophan operon, what is happening? It is an example for repressible operon. Repressive operon. So here when lactose is present, the process is induced, namely the transcription. But here when tryptophan is present, now the process of transcription stops. Now in the case of tryptophan operon, we have regulated gene, promoter, then what we have the operator. But here the number of structural genes 7. There are 7 structural genes. They are responsible for the metabolism of operon. That is what we saw in the tryptophan. Now here the regulated gene produces what is called repressor mRNA. Now this repressor mRNA is called aporepressor, inactive in nature. Here that is what we have, aporepressor. The inactive repressor is called aporepressor. It is unable to bind with operator. In the presence of tryptophan, the tryptophan is acting as what is called actually co-repressor. So this is what is called tryptophan. Now the co-repressor binds with the aporepressor. We have an active repressor. Now this one will bind with the operator side. So here when tryptophan is present, which is acting as a co-repressor. See in the case of enzyme, sometimes some enzymes are inactive. They are called apoenzymes. They can work in the presence of a cofactor or coenzyme to form enzyme complex, or is called holoenzyme, which is functional. Like that, the tryptophan is acting as a co-repressor inside the cell. It binds with the inactive repressor, what is called aporepressor, to form an active repressor. So now we have formed the active repressor. And this one actually binds with the operator region, preventing the transcription. That's why it's an example for repressive operon. This is inducible operon, that's called repressible operon or repressive operon. I'll take in this way, repressible operon. This is the correct word, repressible operon. So, in one operon, inducer induces a transcription. In another operon, now that is a repressor, that is inhibits what is called the transcription when present. So the tryptophan is acting as a co-repressor, binds with the aporepressor produced by what is called the regulatory gene, and both bind together to form an active repressor. Now this one binds with the operator gene and prevents transcription. Hence the tryptophan operon is an example for repressible operon. Another operon what is called histidine operon. Histidine operon. This is also an example for repressible operon. The presence of these substances suppress or repress the activity, but the presence of lactose activates, induces the process of transcription, hence called inducible operon. So this is what is happening, the different types of operons, but you have only inducible operon, lactose. So lactose is an inducer. Repressible protein or repressor protein is produced by the I gene, inhibitor gene, otherwise called the regulatory gene. So the regulatory gene is nothing but the I gene. As it inhibits the process, the letter I is given. Otherwise, you know that one. R will be given, but R is not given. It inhibits the process, the regulatory gene. That's why the letter small letter I is given. Okay. So this is about what we have with reference to the regulation of gene expression.